Hi, my name is Bradley Toller. I'm a postdoc with the SLAC SFA program, and today I'm going to be giving you a brief orientation through the Task 2 poster on microbiology, and I'll be happy to answer further questions and go into more detail during the virtual poster session. So we're going to start by talking about the Slate River field site, and here I have a conceptual diagram of the site showing the spots that we've collected for sediment in the past and in the future, as well as where we're going to start adding pore water. And one of the clear things we see at this site is a distinct transition between oxic and anoxic interfaces. And this happens around 110 centimeters. And this is displayed here through a principal components analysis of geochemistry. And through my expertise, I'm involved in the project to sample the microbial community. And so this is just our brief bioinformatics pipeline here. So far we've looked at the 16S rRNA, which looks at microbial diversity through amplicon analysis. And what we find is that this diversity also aligns with this distinct oxygen transition at 110 centimeters to where the communities in the upper part of the soil column are distinct from those in the anoxic column. And we've also gone into more depth and plan to do more with metagenomic sequencing in the future. But from the outset, what we can see is within these major taxonomic groups, including the proteobacteria, we have key taxa involved in iron and sulfur cycling, as well as nitrogen cycling. And within the archaeal domain, we have some really unique understudied organisms, including the Bathyarchaeota, as well as these big groups of methanogens that can be involved in novel metabolisms and obviously methane emission and cycling, which we're interested in delving into further. Finally, the last parts of this task 2 involve field and laboratory experiments, and these include manipulations to look at different variables and how they impact microorganisms. And so these include a rainfall experiment, seeing what happens to microbes during wet up, as well as batch and column experiments looking at key environmental variables and targeting key transitions, including this oxic-anoxic interface. Finally, we plan to deploy some in situ microcosms to where we can transplant communities during different parts of the soil and monitor after key hydrologic transitions. And that's the poster, and I'm happy to answer any questions for you later. Thank you.